All right, so in theory, you have a genome assembled now. Jacob just told you a bit about how to go about doing that. So hopefully that's a little bit clearer than it was a couple of hours ago. And so now what we're going to do is we're going to take one of the files that came out of that assembly work, and we're going to attempt to predict genes on it. So what are our objectives? To understand the steps that are involved in genome annotation. We're not going to be able to do everything within the, what do we have, 50-minute time period. So there'll be a lot of steps that we'll show the commands for, but we're not actually going to run all of those things. If you're doing real genome annotation on a full data set, it can take, depending on how much data you have, it can take up to a week or more, depending also on the resources that you have. Obviously, if you have a big server and have lots of cores and RAM, you can do things faster usually. But we have a nice little test data set that we're going to use so that some of the things will actually run within the workshop. So yeah, hopefully you'll have some ideas of the types of data and tools that we use in genome annotation, how to quality control the genome assembly and annotation. Usually we'll talk a bit more about this, but usually you want to make sure that your assembly is good enough before you waste all this time annotating it. And then, yeah, just QCing the annotation results. So trying to gauge if you accurately captured the coding regions in the genome. And you'll see that annotation is never 100%. It's you know, you try the best you can to get good gene models, but there's always going to be problems because you're just relying on in silico predictions. So you can get them pretty good, but usually the best annotations have some human intervention involved, manual curation. Okay, so this I'm just showing again to remember that you did this. So everyone should be good to go as far as Docker goes. You did this with Adrian this morning. And this was just to make sure that you had enough room for the files that are associated with Docker. If you didn't move the storage location, you would have quickly ran out of space to store any additional Docker containers. So that's why we do that. And Docker, I don't remember if anyone talked really about Docker, but Docker is a system that's, regardless of what Jacob might think, is usually pretty nice, because what you can do with it is you can take all of the packages and files and things that are necessary to run software, and you put it into what's called a container. So in theory, it's much easier to install for anyone that's tried to install things, or if you tried to install Masurka this morning, you probably realize there's a lot of things that can go wrong. It looks for different libraries on your system. A lot of times those things aren't there. So it can be a bit of a headache. So the idea of Docker is to have everything in this nice little package, and then you just use Docker to deploy those tools. So in theory, you know, it's usually preferable. Like if you're publishing any software, I think that it's really recommended that you supply it as a Docker container, because it's easier for reviewers to deal with the software instead of installing 5 million different things. So that's why we use Docker, in case you've never heard of it before. And I oftentimes, there's something called Docker Hub, so kind of like GitHub, only it's for Docker images. And you can go there and oftentimes find something that someone's already made an image of a tool that you might be interested in for Docker. Okay, so we're going to get this going. I don't really know if we're going to have time to run it, but let's just go ahead and get this going anyways, just in case there is time, or if you want to run InterProScan on your own later. So what we're going to do is I'm going to make this bigger so you can see it. First of all, okay. All right, so I probably have a whole bunch of stuff there that you don't, but you don't need to worry about that. So the first thing we're going to do is we're going to CD into Scratch. So that's where I am right now. And then if you have the slides, so I guess I should tell you, let me me up there. So if you go on GitHub, so everything is here, which has been pointed out previously. So we're on annotation. So the slides are here, which aren't going to show up because they're a PDF. Oh, they are going to show up actually. Never mind, I lied. So the slides are there and they should match mine, hopefully. And then there's a bunch of scripts in here that we're going to use. Okay, so first thing we're going to do is just get this download started. So I already did this. I'm not going to do it again. But basically you just want to use wget, this whole thing. And you just want to download that onto your Scratch directory because it's a pretty big file. So that's why it needs to go there as opposed to opt. I tried to put it there, but that didn't work because there's a lot of stuff in this software. So go ahead and get that going. I'm having a cat battle behind me. If you hear squeals, no one's getting killed, I promise. So go ahead and get that going. And then once it finishes, just as we're going through the rest of the slides, there's some other commands here that you'll need to run on it. So you'll have to unpackage it and then you'll CD into the directory. I guess I missed that step here. You'll CD into the directory and then you'll run Python 3 initial setup.py. So let me, looks like Jing's on top of it there. So yeah, so just go ahead and get that started and then just let it run. And just for anyone that's curious, I'm using, what is this called again? Hyper, which is just basically a terminal. Instead of using the web SSH, just because the copy and paste thing was driving me insane. So you might have noticed that you can actually get an IP address for the image that we're using. So you could just do SSH and then that IP address and then use terminal instead of something that might be annoying for you, or maybe it's not. Maybe it's just me. All right, so the first thing we're going to do is actually do structural. We call it structural annotation. So this is actually finding the genes and determining where intron exon boundaries are, where UTRs are found, that sort of thing. We also need to mask repeat elements in the genome. So this is really important because if you don't do this, repeats in the genome tend to confuse the predictors. And so you'll end up with a ton of genes. It'll think some of the repeats are genes. So you'll end up with a lot of gene models that aren't really, your typical coding genes. So it's always important to do that. And then after we have our predictions, so our coding sequences basically, then we're going to do some things to associate functions to them. So whenever you get a file and it has, functions associated to gene models, usually they're just predictions. In some cases, they're experimentally validated, but usually they're just predicted based on similarity to other gene models that have functions associated to them that again may or may not be 100% accurate. So all of this stuff, if you get a genome file, an annotation that goes along with it, there's going to be things that aren't quite right. Okay. So the first thing you want to do, and so whenever you did your genome assembly with Jacob, the first thing you usually want to do is look at the total length of your assembly. And I think Jacob gave me those numbers. For some reason, my slides don't really make a whole lot of sense here. But so this was the length of one of the assemblies, I think the fly polished assembly. And so this size, you want it to be pretty close to the expected genome size of your organism. So if you remember, Fei Wei talked about some ways that you can get an idea of what the size might be for your organism of interest. So if it's way, way bigger, that could indicate that you have some uncollapsed heterozygosity. So if you're assembling a heterozygous genome, you'll end up with redundant copies oftentimes, haplotigs, due to the heterozygosity. If it's too small, maybe you don't have enough data. And then the other thing you want to look at is the N50 value. So the longer the N50 value, the better. That means you have a more contiguous assembly. If you had an N50 value of like, say you had an N50 of like 5,000 base pairs, do you think you'd want to annotate that? And if not, why? Does anyone have any ideas? How long do you think an average gene is, I guess, in an organism is something to consider. So if your N50 is only like 5,000 base pairs, that means there's a lot of stuff in there that's smaller. So your ability to actually find genes on those contigs isn't going to be very good because you're going to be missing starts and stops, most likely. Genes are going to be found between different contigs and that sort of thing. So the higher the N50 in many ways, the better. But definitely, if you had something really low, it's probably best just to work on your assembly instead of wasting time annotating, because it is a bit of work. And lastly, it's important to check BOOSTGO. So I think Jacob talked a little bit about this. I was kind of multitasking, but I think probably most of you are familiar with this. So in this particular assembly, most of the BOOSTGO set was found to be complete. So these are genes that are known to be single copy, and they should be found in this particular lineage. So I think in general, anything above 90% is complete. Some people are more fussy about it, but we work on a lot of weird stuff, and sometimes the genomes are hard to assemble. So to me, 90% and above is pretty good. Okay, so where are we? All right, so there's a lot of tools you can use. And of course, we're not going to go over them all today, but here's a whole bunch of links if you want to read more about each one of these tools. The tools that have been used in our annotation today are EDTA, and I'll talk a little bit more about that. Repeat Masker, we used Augustus and Maker and BOOSTGO, of course. So there's some other tools here that we didn't use, and not because they're not useful, but just because this is a sample data set and we were trying to go quickly. So another thing is SNAP. So this is SNAP and Augustus are both predictors. So they you have to train them. They have I think it's basically machine learning. So you have a test data set and so you have a training set and a test set, which you'll see later. And you're basically just trying to figure out the optimal configuration for predicting genes in a particular genome. And then Breaker, I should mention too. So Breaker, I used once many years ago when it first came out and wasn't super happy with it, but now it works really good. A number of our colleagues have spoken positively about it, and I know they're using it in their work. And the nice thing about Breaker is that you can just give it a BAM file and that's it. Like all of the stuff that we're going to do today is, you know, some of it you still have to do, but most of it you don't. So you give it a BAM file and it gives you gene models. So it's pretty nice, pretty straightforward. We're just going to do the hard one today because why not? And then you'll know how to do it. Anything else on here? GeneMark is another predictor. I don't usually use that one, but some people do. And then Maker is a prediction pipeline. So I don't know why this keeps changing. It's driving me nuts. So Maker takes some of the other tools and uses their predictions, and then it associates scores to the genes, and then it kind of synthesizes what it thinks the best one is. So you can feed it a lot of information and it tries to come up with the best gene model. Apollo is, so it used to be called Web Apollo, I think it's just called Apollo now, is based off of JBrowse. So if you're familiar with genome browsers, there's a specific one called JBrowse, and you can load your gene models in there. And then what's nice about Apollo is if you're working with a consortium on a particular genome is you can set up accounts for them, and then people can go in and manually curate the gene models. So oftentimes if you look at RNA-seq evidence and how it maps to the genome and compare it to the gene models that you predicted, sometimes you'll see that it didn't get it right. So this allows people to go in manually fix those things. And then you can dump that information from the database and incorporate it in your next version of the annotation. Okay, any questions about that so far? Take a minute to catch my breath. I don't talk so much anymore since the pandemic, so I may start losing my voice at some point. We'll see. Okay, so this is kind of an overview of what we're gonna do. We're not necessarily gonna do all of this hands-on today, but it was done at some point in the process. So whenever you have a genome, the first thing you wanna do is the repeat masking. And we typically use these tools for the repeat masking, and I'll go into that in more detail and whenever we look at the scripts. And then the other thing that we do is we take RNA-seq evidence, for example, and that's all mapped to the genome. And that information is used in the predictions. So you want to have a lot of RNA-seq is great, especially if it can be from different tissues, because you might have some RNAs that aren't expressed in some tissues, but are expressed in others. If you have treatments with pathogens, all that kind of stuff can help improve it. And to map the RNA-seq, we use HiSat and StringTie. And then we clean it up with two tools, Portcullis and Mikado. So once we have the genome repeat masked and we have all our RNA-seq mapped to it, then we go ahead and run Maker once we have that sort of pre-work done. And then Maker is what will take all that information and attempt to give you some nice gene models. And when you run Maker, you can give it protein evidence too. It will just map that on its own, although sometimes I've found that it can do some weird things where it merges gene models. So if you have like tandem repeats, sometimes it'll screw that up and make just one super huge gene model. So it's just something to be aware of. Okay, then once you have your gene models, usually you need to do some sort of filtering on it. And that really varies by genome. Sometimes you get the number that makes sense. Sometimes you have way too many genes and then you have to figure out why. Sometimes you don't know why. So there's some different steps you can use to try to get rid of things that might not be real genes. All right, and then you just take your genes and use Diamond to match them against some different databases, and then you can use InterProScan to associate Go terms and domains and all those sorts of things. So this is something, I'm going to show you the code, but I don't think that there's time to do it. We have a file prepared for you. But this is EDTA. So it's a pipeline that basically gives you a set of TEs that you can then use with Repeat Masker for masking the genome that you're about to annotate. So you can see it includes a number of different tools in here. And basically your output, do I have it on the next slide? I guess we'll see it in the shell script. But this output goes into two later steps that we'll go over in a bit. So it will look, one of the things that happens is repeats can be identified by kind of like coverage. And that gets a little bit tricky, and I'll tell you more about that in a minute, but that's how some of these things are working to find de novo repeats. Okay, so I think I mentioned most of these. Mercator is another tool that you can use for functional annotation that you might want to look into. And then these are some of the databases that we usually use to blast the genes that we get from our annotation against these databases to try to give them some functional predictions. So SwissProt's very highly curated, but you might not get as many matches as you'd like since there's not as many genes there. Tremble's another good one. These are less curated, so it may or may not be right, but you'll get more information that may or may not be right. You can use NR from GenBank, and InterPro is just looking at domains again. All right, so now let's do something. Where are you going? Okay, so this was the original assembly file, And then what we're going to do, because there's no way we could do all of this in half an hour. I already ran the repeat masking and I'll show you how that was done. And we're only going to look at one config. And I forget how I selected this config, but I think it was probably one of the mid-sized ones. So we could be sure there'd be some genes on it. And then there's some data in the SRA that we've already gathered up for you to do the annotation. And then we have proteins from uniprot plant. So these are curated proteins, and we'll use those with Maker to help improve the annotation. And hopefully, if your copying went okay at the beginning of the workshop today, all of this stuff is here on your virtual machine. And I think for most of these things, if it doesn't work for you, we should have a copy of it in here so you can just grab it from somewhere else, which is actually what we're going to do in a few places. And I better get moving. I only have 30 minutes. Okay, so let's do this. So hopefully you're already in Scratch from earlier. And if that Interpro, if you're downloading Interpro scan and it's still going, you can type Control-Z and that will stop it. And then you can type BG and that will put it in the background. And that will allow you to get your command prompt back so you can type commands into the terminal again.
Susie, just to be sure, you have an hour left.
Oh, I do. Oh, awesome.
You're okay until 3. Yeah.
Woo, thank you. We might actually get somewhere then. All right, so we want to make sure we're in Scratch. And then we're just going to take our Git repository and we're going to clone that. And I already did that, so it's not happy that I'm trying to do it again. But if you do that and you don't already have that directory there, you should be good. And then let's just check out that directory a bit. So this thing here is just the same thing as this, which you'll see. So see, there's all our directories. There's all our directories. So they match, which is good. If they didn't, I'd be confused. So if we go into annotation, which is the section we're on right now, you'll see there's some scripts in there. So these are all shell scripts. If you're not familiar with shell scripts, they're just basically little text files that you can execute that contain a bunch of Linux commands. So you can either look at them here, like that. So Q for less to get out. So that's what I did. And then Q to get out for quit. And then you can also look at them over here. So annotation, scripts. And there it is. So this is, you may have noticed, just feel like this is so clever. They're numbered in the order that they go. That's probably obvious, but hopefully it makes things a little bit easier. So the first one we're going to look at is repeat masking, because we just established that that's the first thing we need to do once we have the genome assembly. And we looked at this already, so on to the next one. Okay, so step 1 is repeat masking. And this was already run, but we're going to look at the shell script to see how it was done. And in theory, these should work if you try to run them on your virtual machine, but I think there was one that was giving me a problem. I don't remember which one it was now. But if you want to try, you can go ahead and do that, of course. So the first thing it does is it uses Docker, and it pulls in the EDTA program. So that's the one that's going to predict the transposable elements. And then it goes into a directory, which I think maybe hasn't been created yet. So you'd have to make the directory. So all of our work is going to go in here today. And the next script actually creates that directory, so you don't need to worry about it right now. So then the next thing is it runs EDTA on the Polka corrected fly assembly. And it uses 7 threads. So I think that's okay on our virtual machine. If you're running this on a big server, you'd probably want to crank that up more so it runs a bit faster. So then the next thing we do is we want to see if any proteins, any known proteins, so these are known proteins from UniProt again, if any of them match this file. So this file is the output from EDTA. And what sometimes will happen is, as I said, since the de novo repeat prediction is somewhat based on kind of the frequency of certain sequences, you know, if you have transpose, like repeat elements, there's going to be a lot of them, right? So something that's a very high copy number could be a repeat. But the problem is that sometimes you'll have protein domains, and those can be rather high copy number as well. So the prediction might call things that are legitimate proteins repetitive elements. And then whenever you mask your genome, they're all going to be masked, all those sequences. So whenever you try to do the annotation, you're going to have proteins that are missing probably pretty important parts for their function. So you don't want that. So that's what these next two steps are about. So we find anything in our repeat set, this consensi.fa.classified, and we see if any of that matches known proteins. And then if it does, we decide we don't want to mask that. So we remove that. So that's what prot excluder does. So once we have prot excluder output, then we can go on and run repeat masker. So repeat masker is the one that's actually going to take the genome, and then it's going to mask it. So in this case, we do hard masking. There are some more involved ways of repeat masking, but we just do it kind of simply for this workshop. So we use our, this is the file that comes out of prot excluder. So this is all of the things that were predicted to be repeats minus anything that looks to be a protein domain. And then this is our assembly that we want to perform the masking on. So when you mask, there's hard masking, which will take anything that looks like a repeat and just make it ends. And then there's also soft masking, which will take just the letters and make them lowercase. So for this effort, we're going to use hard masking, which I think actually is output from a repeat masker. There should be a GFF file, I believe, and a FASTA file. But another way to do it is you can take the GFF file here. So a GFF file is just a tab delimited file that tells you where things are found in the genome. So we'll generate one of these for the gene models too. But in this case, this is just telling us where in the genome we seem to have things that look like repeats. So it'll just take the GFF and then the original assembly file, and then it'll generate, oh, I know why we did it this way. That's right. I must have taken it out beforehand. Yeah. So we're actually just masking one of the contigs. So of course, if you were doing your real project, you wouldn't do it this way. You would run this on the whole genome. But for a test data set, we're actually just going to look at this contig 15. So just one contig out of the whole thing. So as you can guess, our final the results aren't going to be great, but it allows us to at least try some of these commands out. Yeah, so that's basically it, just making our input data. So any questions about that?
Where have you got this NMG Workshop 2021 folder from?
Do you mean the git clone command?
Yeah, was that supposed to work? Like,
oh yeah, it should work for everyone but me, because I already...
how long is it supposed to take?
It shouldn't take more than a couple of minutes.
Oh, it seemed to be saying like it was going to take half an hour or something.
Huh. Yeah, when I ran it earlier, it was pretty quick. It was just a couple of minutes. I would just let it run. You can put it in the background with Control Z and then type BG. And
wait, what?
So type Control and then the Z key.
Doesn't that end it?
that just stops it. And then when it stops it, it brings your command prompt back. So you can type BG for background, and that allows it to run in the background, but you can still type things on the command prompt.
What? So so if I typed out, it's nothing's happening at the moment. If I just type out that thingy, whatever it is, command,
Mhm.
the thing,
Control Z. Yep.
the we get F, is it the we get FT? Well, no.
Does
What was that first? We get FTP. FT
maybe the
multiply
interpro
massive.
scan? Yeah, let's go back to that.
Was that supposed to work?
Yeah, I hope so. So this was the first command. So this is for downloading interpro scan, which was too large for me to get onto the virtual machine prior to launching it, unfortunately. And then after it's done downloading, then you run this. It'll take a while to download because it's pretty large. And then here it should be, I noticed someone is telling me about the problem. You'll have to CD into that directory before you run that command. So for Sirin, I think what you need to do is just type CD and then interpro scan the name of the directory. And then once you're in the directory, yeah, if you run that, it should hopefully work. That downloaded pretty quick, quicker than it did for me. Yeah, so are we good?
I'll give it a bit. Thank you.
Where was I on my slide? Okay. All right, so just feel free to shout out if anything weird happens and we'll try to get things on track. Okay, so the next thing we're going to do is we're going to map the RNA-seq to the genome using HiSat, and then StringTie is going to give us some ideas about what potential regions might be genes. It kind of gives us some rudimentary gene models that then Maker works on to make them look a little bit better. So let's look at that. So in this one, I think we can just go ahead and run, and we'll see how that goes. So if you want to get this going, we'll do that first, and then I'll explain what it's doing. So if you want to run this, I'm not sure if I put it in the slide. Looks like I did not. So you should just be able to type. So let's go into annotation. So now I'm in the annotation directory. And then I want to run the shell script. So if you did the git clone of the NMG workshop 2021 from GitHub, you should have this directory. And if you go into annotations and scripts, and not there, and so we want the HiSat PE annotation.sh. So does everyone see that okay? So I'm going to go ahead and hit enter, and then just maybe sort of keep part of an eye on it to see if anything weird happens for you. But you should just see a whole bunch of stuff that goes flying by your screen like this, ideally. If it does something and then quits right away, that's probably not ideal. And I did get this error message previously. I'm not sure why that's happening, but it did run to completion for me. So we'll see what happens for everyone else. All right, so let's see if I can get back up there. All right, so this everyone, and then cd scratch. Actually, scratch that. We want to do this. I'm confusing myself. Scratch. Anna. Actually, it doesn't matter where you are when you run it. So you can just run it like that. So there's a space after bash. And I think regardless of where you run this thing, the files should end up where they're supposed to be. So I'm going to put mine in the background. So this is what I was talking about before. So you can type Control Z and then background. And then see how I can type things now? That's not an actual command. There we go. So now I can type again. And if you want to see if things are still running, you can type top. And SAMTools is running. So that's probably part of the script that I just launched. So that's a good sign. All right. So okay, so what is this script doing? So as I said, a shell script, for anyone that hasn't encountered this before, is just a program that in this case is using bash programming language. And you can make them really easily just by typing a bunch of Linux commands into a text file. So they're nice because you can use things like loops to automate things, which we do in this script. And I guess the hope is that, you know, if you're someone that's not already doing a lot of programming in bioinformatics, that some of these things maybe you can reverse engineer for your own needs. Okay, so what happens here? So the first thing it does is that it makes this directory, scratch annotation output. So if you notice, scratch, you should now have a directory there called annotation output. So I'm in that directory. If you want to go there, that's where all the output from the script should end up if things go as planned. So it makes that directory and then it CDs into the directory. And then it copies a file. So this config 15, you might remember that from the repeat masking script. This isn't repeat mask, this is just the config pulled from the assembly. And we're just using only that because otherwise this would run way too long. But obviously, if you were doing your own project, you wouldn't want to annotate it on a config by config basis. That'd be really annoying. So just keep that in mind that this isn't really a real life thing here. And then the other thing we do is we symlink into that directory. So this is a symbolic link to FASTQ files. So symlinks are just nice because you're not making a redundant copy of the data, which would take up disk space, but it does kind of make a placeholder for that thing. So for all intents and purposes, it looks like these FASTQ files are in our working directory, but they actually live somewhere else and it's just a pointer to where they are. So it just saves disk space, which is nice with this type of stuff because oftentimes you'll end up with enormous files. Okay, and then I should say too, any of these things you can just type in, like you can just copy and paste that into the terminal. They're just Linux commands. Then the next thing it does is it starts using HiSat to map the RNA-seq. So anyone that's done RNA-seq before, you're probably familiar with this tool. So the first thing you do is you index the genome file that you're going to map everything to. So that's what's going on here. And this just allows the mapper to kind of figure out where reads match in the genome. If you don't do this, HiSat will complain and it'll crash. Okay, so the next thing it does is a loop. Loop-de-loop. So here's our loop. That whole thing's a loop. And in this case, it wasn't really so necessary because we only have 2 files. But again, this is just like, you know, if you're not familiar with this stuff, this might be something useful for real data. So we wrote it in a loop because oftentimes, most of the time, I'd say you have a ton of RNA-seq data that needs to be processed at once. So this just will go into a directory, look for all the FASTQ files that it finds there, and then it'll map them. And in this case, we have paired end data. So it actually goes in and it finds the mate too for that FASTQ file. So it's just creating some variables. So this is a variable for the output file name, and then this is a variable for the second file in the mate paired end mate. So it creates those read names, and then here's where it actually runs HiSat. And it just uses these variable names that we created up here. So you use these variable names so that you have a different output file for each input file that you have. Otherwise, you're just going to overwrite things as it goes through. So it's important to have a different output file name in these loops. All right, and then it just does some file conversion. So anyone that does a lot of bioinformatics knows that you spend a lot of your time either Googling stuff or reformatting things from one file format to another. And whenever the files are really big, this can be kind of time consuming. But in this case, it's not, it's pretty short. So in this case, we only have one SAM file. So the SAM file is the output of HiSat. So this is telling you how all of your RNA-seq reads are mapping to the genome. And then it uses this tool called parallel. So if you had 7 SAM files, let's say, then what would happen over here is you would have 7 instances of SAM tools running. So this would allow it to run faster as opposed to just running on one core, one instance. So it sorts, this isn't the sort. So this is taking SAM to BAM. Does anyone know why we convert SAM to BAM? I'm sure there's some of you out there that are familiar with this stuff. Silence. Okay. Oh, computers like it better. Yay, Andrew, bailing me out. Yeah. So one of the really good things is that SAM, BAM is like a compressed SAM. So it's more efficient, it's better on disk space. And just generally, most of the downstream packages you'll find want things to be in BAM format. So once you have it in BAM, you can essentially delete your SAM files and it'll save you some space on your machine. Okay, then it takes that BAM file that it just made from the SAM file and then it sorts it. So this is just sorting the reads based on coordinates. So the first read on the first chromosome is at position 1. The second read on the first chromosome is at position 2, something like that. So it just sorts them in that order. And again, you might find that this file size actually shrinks a little bit after this. And also most of the downstream tools expect it to be sorted by coordinate. Sometimes things should be name sorted, but that's kind of rare. You just have to read the documentation for whatever you're trying to do. All right, and then we have our sorted BAM. So again, in this case, it's kind of funny we're using a loop because it's just one file, but this is just, you know, something that may help you in the future when you have a lot of files. So we have another loop here, and it just finds all of the sorted BAM files. It creates an output directory, and then it runs string tie. So string tie takes the BAM files, so those files that tell you where all of the reads are mapping in your genome, and it converts it into a GTF file, which is a text file, so you can just look at it with less or something. Actually, we'll look at it in a minute. And this gives you some idea of things that might be gene models. So it takes all those messy BAM reads mapped to the genome, just all over the place, and then it tries to arrange them into things that actually look like intact gene models. Okay, not a breath again. So I think mine is done. It says it's done. That's promising. Thumbs up if other people's seems to have completed. Nice. I see some thumbs, although I need to make this bigger because I can't see most of you, I'm realizing. You could be making horrible faces at me and I would not know. You probably should make horrible faces at me. Okay, cool. Now I can see everybody. Or everybody's boxes with their names inside. Okay, so if you type, so this is where I am. I wonder if I should move this up a little bit, see if that makes it easier to see. So this is where I am right now. I'm in /scratch/annotation_output. And if that script did it was supposed to do, you should see something like this. You're not going to see this. Ignore these folders at the top. That was just me messing around. But the rest of the stuff you should see. So these are the FASTQ files from Utricularia, traps and something. I think I have it in my slides, but I don't remember now. Shoots and traps. RNA-seq from shoots and traps. So those are the FASTQ files, and these are our index files here. So these are all the index files for the reference genome, which is really a contig. Here's our CM file. So you can see the CM file is 1.6 gigs. But then when we convert that to BAM, it goes down to 411 megs. And then we sort it and it gets even smaller. So if this was a real project and you're, these files would probably be much bigger, you could just go ahead and get rid of these two. Assuming this, sometimes you'll have a truncated file and then you'll be mad you deleted all those, but usually it's safe to get rid of those to save space on your machine. Okay, so now we have this file that has all of the BAM, all of the reads mapped to the genome. And then StringTie generates this thing for us. So this is a GTF file. Let's look at that. So you can use less and then the name of the file. So I'll try to make it a little bit bigger. Actually, type Q and get out of there. Let's do this the right way. So we're going to use an option so the fields line up a little bit better. So it should look like that. All right, so there's our GTF file. So this is what we're going to be using for the next part of the RNA processing. And you can see, so this is telling you which contig things map to. So it's going to be contig 15 for everything because that's all we had. But if you're doing something real, this would be all your chromosomes or contigs or whatever you have over there. The software that was used to predict it. And then what the feature is at that particular. So this is saying we have a transcript on contig 15 at 10,631 to 13,491 base pairs. And it's in the plus orientation. I think this is some kind of a score. I forget how that exactly is assigned. And then we have some names. So these names don't really mean anything right now, but later on, once you're getting your final annotation file, you usually change these into something that make a little more sense. So like if you work on tomato, it's a Solic IDs, or if it's Arabidopsis, it's AT something. So these are just kind of the messy StringTie ones. And then something to keep in mind for later is it also gives you FPKM values. So this can be useful for filtering once you have your maker output. So sometimes people like to get rid of features that have very low expression because those might not be real genes. So this is good information to have too that we don't use initially in the annotation, but can come in handy later on. Okay, so does anyone have any problems so far? Cool, I don't hear any complaints. So we'll keep going. Keep losing myself in my slides here. What am I doing? Okay, so now we're doing step 3. This one, I think we're probably not going to go through all of the steps. I think we'll just talk through this one. But essentially what we're doing here is we're taking the RNA-seq output from StringTie, so that GTF file. Actually, I need to look at the script. And it cleans that up because a lot of times RNA-seq is usually pretty messy. So these two tools, Portcullis and Mikado, use some algorithms to try to just clean them up and make the gene models that result from them a little bit more accurate. And there's various ways of doing this. This is just what we've found has worked well for us. But not everyone that's doing annotation is going to use these same tools. So let's go here. I think most of this should work, but then I know there was a problem down here. I think the HMM scan was problematic, and that actually runs a really long time too. But if you do want to try running some of these, you can. So I think we'll just talk through this one though, just so we can get through everything. So, I mean, if you really wanted to have some fun, you could run the script and see what happens. But I think when it gets to HMM scan, it's going to be angry because I think there's some problems there. But any of these, you can just try to run. Okay, so what's going on here? So here, we're getting some files that it needs to run the tools that are in this directory that you copied over earlier. So those are all ready to go, and they're found there. Then it uses Docker to install Portcullis. And I think you already did this with Adrian. So in theory, this should work. Let's see if we can get it to work. Let's just give it a shot and see what happens. I can actually copy the thing. Okay, so that seems to work. All right, so what Portcullis is doing is it's taking our BM file, and I guess it doesn't use the GTF, I had forgotten. So this is just taking the BM file and cleaning it up. And then it outputs the cleaned up file into this directory here. So whenever this is done, we should have a new directory called SRR, whatever, config 15 port for Portcullis. So this may take a while to run. So if you just want to let that go, you can, or you can kill it or whatever you want to do. So the next thing that's going to happen is we run Mikado. So Mikado is going to take the output from Portcullis. And I think in this case, we also use the string tie output. Let's just look real quick. See if I can put this in the background. Doesn't seem happy with me. I might have to kill it. I'm killing it. Bye-bye. So this list file, so we're right here, is just a list of the output that you got from string tie. So it's sort of like our pseudo gene models. So it takes that, it takes the reference genome, which in this case is just the config 15. And then it takes some other parameters. And then it also takes the junctions file that is output from Portcullis. And then you can also give it the Swiss proteins as well that it will use as sort of like validated gene models. And so it takes all of that and then it creates a configuration file. So this is sort of like the configuration file for the whole pipeline. Then the next step is it takes the, so these steps up here create a file called Mikado prepared FASTA. And that's just sort of like its first guess at what some gene models might be based on your input data. So based on that GTF file and the BM file. So once you have that file, then it runs BLAST against your UniProt database. And it generates some BLAST output that then you use later on in filtering. Some of these steps are optional too. So if you look at the documentation, you don't necessarily need to run HMM scan, but I think that in general, it just gives you a little bit more certainty about whether those predictions are likely to be real coding genes. So they're good to do. So transdecoder is just getting the longest open reading frame from the first sort of prediction. And then it's also running HMM scan. So this is sort of looking for any sort of domains that match known proteins as well. So essentially all these steps are just doing comparisons of your genes to known genes to see if what you predicted as genes actually should be genes. And then it's doing some corrections so that you can get a more accurate gene model. And so the reason this is really nice is because not only does it kind of give you a first pass annotation, it misses a lot of stuff because you're only really basing this off the RNA-seq. But the output is usually pretty accurate. So what's nice about this is it allows you to come up with a training set for the later steps of the pipeline. So whenever you're training Augustus, SNAP, or GeneMark, the output of this gives you a nice set of genes that are fairly accurate that you can then go on and use as training sets. So see if there's anything else I wanted to say about that. So the end file from all of this is this Mikado. It actually gives you a few files. So there's Mikado loci.gff3, and then there's Mikado subloci.gff3. And this file actually gives you a little bit of scores associated with the genes so that you can try to pick out ones that have a higher score that might be better for use in the training set. Okay, get out of here. Go back to the slides here. All right, so all right, so once you have the output from Mikado, then you can kind of get to like the good stuff, I guess, where you're actually running Maker and getting some real gene models. So these are the three probably most commonly used prediction tools. And today we're just going to look at Augustus because it tends to be the one that's more tricky. I think in the slides, yeah, here, if you go to this link, you can get more information on how to train SNAP, but SNAP's pretty easy to train. So, and then GeneMark, we're not going to use that one today, but it's something worth considering. All right, so yeah, we're just running the second script, and then we're going to skip the third script because we have some output already generated. It would take a very long time to run it, but you can try running it on your own. So now we're going to go on to the fourth script. So we're going to use that script to try to train Augustus. So let's, the little thing keeps coming down. So let's go to scripts and training. Okay, so let's try this. I think you can do this one of two ways. It's entirely up to you. You can try to run it. So if you wanted to run it, you would just run it like that. I'm going to go through and actually do it line by line. Actually, no, I'm not, because there's one step that takes a long time and we'll be waiting a while. Okay, let's just run it this way. And then while I talk, hopefully this will catch up to us. So yeah, oops, what did I do? Something weird happened. Okay. Oh, maybe that portcullis is still running in the background. All right. All right, let's see what just actually happened there. Oh no, it's just doing something weird. Okay, I guess we are going to run it line by line. I don't know what's happening. I'll have to figure that out later. That's annoying. Okay, so the first thing we're going to do, you can just open this up and follow along. So this is the training script. Okay, so the first thing to do is make sure you are in the annotation output directory. And try to resize things here. Okay, 20 minutes. Yeah, so make sure you're here. That's where I am. And then we're going to cheat. As I said, that last script would take too long to run. So we're just going to grab that file from somewhere else. So that's our Mikado output. And we can take a look at that once it copies over. So let's take a look. All right, so you can see this has maybe a little bit more detail in it than the GTF from StringTie did. We have things arranged into genes now, and mRNAs, exons, we have UTRs. You're not going to have them for every gene, but yeah, I guess it just depends on what the data look like. And then coding sequences and exons. So I think we could probably, let's see. Oh, one thing, so that business with Uniprot, whenever we were looking at the Mikado script, it's actually assigning things as genes that had matches to Uniprot. But if you go through here, I think there's like non-coding RNAs or something. I'm not seeing it here. Let's do this. Cut F3 Mikado and sort. Okay, so all I did was I took the third column and then I just wanted to see all the unique things that are in there. Yeah, here we go. This is what I was looking for. So whenever it finds something based on your RNA-seq that looks like a gene, it matches it to whatever database you tell it to. So in our case, it was Uniprot. And if there's not a match there, it tends to just call it a non-coding RNA, which is a little weird because it could really legitimately be a gene. But just something to know if you're using this tool. I guess we can even do this. See how many of each thing we have. So it looks like we have 1,703 genes. And of course, more exons and coding sequences as you'd expect. And it did a fair number of UTRs, actually. Cool. So just, yeah, keep in mind, that's only one contig too. So that's not the whole genome. It's just contig 15. All right. So I guess we kind of did this next thing, but if you want to look at it a different way, you can just find all of the genes by using awk and probably get the same number. It should work out the same in theory. All right, so what we're going to do now is we are going to take our Mikado loci and we're going to convert that into protein. So I'll explain what I mean here. So GFF read is a tool that will take a GFF file. So in this case, our Mikado output, our predictions, and the genome file, contig15.fasta, and it will give us a FASTA file, the protein sequences that we need for later things. So you can go ahead and try that. And if that worked, we should have some proteins. So there we go. So it looks like most of these start with methionine, so that's good. And I guess it's not really giving us the stop codon. It's probably just cropped out. But there, that gives you an idea. And let's see how many we have. It's probably just going to match what we had before, but let's just see. We'll see. Ah, it's actually a few less. I'm not really sure why that is. Maybe, no, not sure. It could be something related to isoforms. I'd have to look into that. But however many genes we had in the GFF3, it seems like they're not all showing up in the protein file for some reason. Something to investigate later. All right, so now we're going to run OrthoFinder, which seems maybe kind of random, but I'll explain why we do that. So we're going to make a directory for the OrthoFinder input. And then we're going to do this sort of hack to get what we want. So we're going to move our protein file into the directory that we just created. And I don't usually copy and paste everything, but I don't want to, things are already confusing enough. I don't want to start typing things wrong and making it worse. And then we're just going to copy that file, make a copy of it in the same directory, basically, is what we're doing. And the reason we do this, does anyone have any ideas why we do this? Because there's various ways of approaching this problem, but this is a way. Does anyone know what OrthoFinder is? Probably not.
I'm
Yeah.
guessing it finds the gene. Can you still hear me?
I think I lost you. Can you repeat that?
Oh, right. Can you hear me now?
Yes.
Oh, fab. Yeah, search for the orthologous genes from, I assume, the reference, if we're using a reference, from probably a different species, if this is constructing the new. So it will find whatever ones match that will be related.
Yep. Yeah. So that's how it's normally used. So that's why I'm saying this is kind of a hack, because normally you'd be looking at orthologous genes in different species. For this purpose, all I wanted to do was arrange things into gene families and try to get rid of redundancy. One of the problems with training is that if you have too much of the same kind of gene, this can result in overfitting of the model. So usually, if you read the documentation for Augustus, it recommends that you keep proteins at like less than 80% identity. You should have gene models that represent the distribution of like number of exons per gene that you find in the genome. I mean, I guess it's kind of hard to know in advance, but you just kind of want to get a representative sample. You don't want a lot of single exon genes, because then you're not going to be training the intron exon boundaries.
So will this be matching the genes basically with themselves?
Yep.
It's all still internal, but you're just going, oh, which ones are likely to be in the same family?
Yep. Yeah. It's making gene families with itself, and just, it doesn't let you do it with one file, which kind of makes sense, because ordinarily you'd be looking at different species. So it's just sort of fudging it. Yeah, so that's why we do that. But you can also do other things, like you could do some sort of clustering based on sequence identity and just remove anything that is redundant, so that you just have one copy. Yeah, but we're going to do it this way for today. Okay, so where was I? I was here. So let's run OrthoFinder and see what happens. Here we go. This is a really nice program if you're interested in comparative genomics. It will even generate alignments for you. It'll generate gene trees. And then you can use those alignments to go on and do more, I guess, rigorous types of analyses. So it's, what we're using it for isn't really showing off of all of its strong points. Okay, so while that runs, I think it should run pretty quickly. All right, it's done. So the next thing we're going to do is we're just going to get a gene from each group. So let me show you what I mean by that. When am I done again? At 3 or 3:30?
3.
3. Oh, I thought I had so much time, and now I have
original
none.
schedule.
How did that happen? Okay. Let's focus on what I'm doing here. Where is it? Okay, let's just do this. I'm getting flustered because I'm trying to hurry. Okay, so this file, if you didn't see what I did, I just kind of copied that whole deal because I was being too silly to type it out. And so this is, if you go into the OrthoFinder results, there's a whole bunch of stuff in there that you might want to look at later if you're interested. But we're just going to use the listing of the ortho groups. So that's what this looks like. And again, let's use a better version of the less command. So the ortho group ID is on the left. And then here we have all of the genes that are part of that group. So by doing this command here, we just get one gene from each group. So it should help to prevent that overfitting problem of the model that I mentioned. So let's go ahead and try that. So now we have a file that's just a list of genes These don't really look like gene names, but that's how they look at this point in the pipeline. All right. I might just walk you through the rest of this because we're getting kind of close to time here. So this git clone is just getting a nifty little tool that is used in line 27. First, we need to get the file names converted correctly. So this is just doing some work to that protein file so that it's going to match our list that we made up here. So that's what that's doing, just some finagling around to get things to work and be in agreement with the naming structure. Okay, so now the git clone, we got this tool, so let's use this tool. So this is a nice tool you might want to use for some other things that essentially you give it a list of sequence IDs and then a file to extract those sequence IDs from. So that's what happened here. We have our list of genes that we want to use for training, and we're just getting the sequences for those genes from our original FASTA file. And then it's making this file down here. So those are the proteins that we're going to use in training. All right, so this step takes a while. So I think while this runs, I'm just gonna talk you through the rest of it since we're getting close to time here. But this is probably gonna run a good 10 minutes anyways. So once this finishes, so this is basically taking your protein. So this is gonna be your training set. So that's what we just got from up here. From the FASTA extract tool. So that's going to be your training set. And those are some pretty high quality gene models that we're reasonably sure are mostly correct. There's always going to be some problems, but they don't have to be perfect to be training sets. They just, you want them to be as good as they can be, but nothing's ever going to be perfect. So we're taking those and then we're mapping them back to the genome with this Scipio tool, which essentially runs BLAST. And then we run a series of commands that I unfortunately kind of have to gloss over here. So once we have the mapping, then it generates a file of genes in GenBank format that are used for training. So let's go. So we were right here. So first it generates a GFF file. Then it does some things to, here is the part I'm talking about. So here's where you actually end up with some GenBank formatted gene models. And the reason you have to do this is because Augustus wants things in GenBank format, which I don't really use that often, but I know is another popular format for these types of things to be in. So you need to basically figure out how your proteins are arranged on the genome and then get that into GenBank format. So this is getting back to bioinformatics kind of moving a lot of file formats around. Okay, so then you do some training. So you do this training, which kind of gives you an idea of which gene models look good and which look bad. So anything that has an internal stop codon, or maybe it doesn't have a start and a stop, those are what go into this file called bad genes. So you're basically figuring out which proteins don't really seem to align to the genome properly, and you're getting rid of those from your training set. So once you do all of this, this is your final set here. And where's the split? Why am I not seeing it? So you filter out those genes that look bad. And then here we go. This is the part I was thinking of. So this script here will randomly split that file of pretty good gene models into your training set and your test set. So that's what these next steps are. You're essentially making a model for Utricularia. I knew I was going to do that at some point. I just could not say some words. So you create a new species, and then you do your first round of training with your training set. When you do training, you usually want to have at least 200 genes at a minimum. More is better, but there is a point where you have diminishing returns. And so you do the training, and this creates some configuration files based on your genome that should give you some information about sort of how a gene looks in this genome, so that it can try to use that information to more accurately predict genes. So then that's basically the last step here. Is it takes the test set and then it sees how good it performed. So I have some example output we can look at here. So whenever this runs to completion, you'll get a file called firsttest.out. And this was from a previous run. And this gives you some idea of how accurate the prediction was. So you have the nucleotide level, the exon level, and the transcript level. And you can see at the gene level, it gets not so great. So like basically each one of these is a little bit more complicated to get it right. So nucleotide, like this would be acceptable for a genome project. And I think for exon level, this could be a little bit higher. But the gene level is always like, I think the best I've ever able to get down here is like 0.5. So in this case, you're not really getting any of your gene level predictions correct. So you'd probably want to go back and maybe do some manual curation on that training set, or maybe try another way of making a training set. So you could look at sequence identity instead of maybe using OrthoFinder. Every genome's different, and I've found that what works for one genome doesn't necessarily work for another. So basically you just work on tweaking things until you get something that's acceptable. Yeah, so I think real quick, we're just gonna look at these last couple of steps. And then if you're doing this with your own project, you can feel free to reach out to me and we can help you customize these things to what you need. All right, so 4 minutes. So now we're here. So we did the repeat masking, the mapping, the cleanup. We did the training sets for Augustus. And now the next thing would be to run Maker, which, as you can probably guess, takes a bit of time. But we have the output in the NSF and FS mount. Okay, so for Maker, let's just try this so you can kind of get a feel for what Maker looks like whenever you're running it. Why is this frozen? Okay, so to get the control files, you can use this command. So Maker generates, I think, 3 text files that contain information. One of them is for the general configurations, like where BLAST is, where different tools are found. One of the files that we'll look at real briefly is where you actually put your input and which genome you're going to annotate and how you want to annotate it. So if you run, oh, I know why. Probably have to put the whole path. The way that we install things on our servers are not the way things are installed here. So can be a little confusing. Here, does that work? Okay, I think that worked. Okay, so now we have, oh, I guess there's 4 files now. Used to only be 3, I swear. So these are all the control files. And you can look at the other ones, but the main one is this one here for establishing your input files and all of that stuff. So I'm going to use Emacs and we can take a quick look at that. And so you can see it's just a text file, but this is the input file that directs Maker to everything. So you would put your mass genome here. And I think I have this information in the script. So these are the things that you'd want to edit in our example. So you'd want to add the mass genome, and then you can add the curated proteins under protein. So down here, you can see it takes all sorts of information. So if you had some EST sequences, you can throw those in. If you had some aligned sequences in GFF format, you can put those in. It'll even do the repeat masking for you, but I usually do this, or I should say Adrian usually does this using the pipeline we already discussed and not running it through Maker. And then you generate some model files for SNAP. So this is the SNAP HMM model. If you're using GeneMark, that would go here. And then this would be your Augustus configuration. So after your training, you would just add that so it knows where to find that information. In our example, we call it UGIBA, like that. And then you can also use that Mikado output as well under predictions to just sort of guide Maker and some other independent predictions outside of the Maker pipeline. Okay, so we can get out of here. Maker has pretty good documentation too, luckily. There's a lot of and then to run Maker, usually one thing that's really good is to use MPI run. So this allows you to run the command on a number of cores. If you don't do that, then you usually just run it on one thread, and it can take like probably weeks, if not months. I never tried it, but depending on the amount of data you had, that would not be ideal. But for a course, you could just use this command. And then I guess maybe we can look at this. I'll show it to you, and then if you're interested, you can look at it more in the future. But here's the output. So if we'd actually ran the entire command, we would have something that looks kind of similar to this, so we did some other things here. So once you have your Maker output, you have this data store, and then you have this log file. And you can use this GFF3 merge command to generate the GFF3 file that'll have your gene models. So let's see what that looks like. I already generated it previously. Oops. Try this. Yeah, and there you go. And you can see there's still some ugly names in here. So there's a script that comes with the Maker package that will take these and turn them into, I guess, more user-friendly gene names. But this is essentially what your output would look like. This is just a pretty standard GFF3 file that tells you where all the gene models are. And then the last thing you'd want to do once you have this, so let's look at the command again. So you can use GFF read again with your GFF3 file and the genome file to get a protein file, a protein FASTA file, and then you can run Busco on that. So that would be the command down here if you wanted to do that. And that will give you an idea of how good you are able to actually annotate genes. So if you know that the Busco for the genome is 93%, then if your annotation was really accurate, then you should have a number that's pretty similar when you run it on your protein set. If you know that you have 93% of the Busco genes in the genome, but you only annotated 60% of them, then you probably need to work on your annotation a bit more. So that's one of the ways of QCing the annotation. Okay, I'm 2 minutes over. Last thing. Let's see if there's anything more on the slides, really. So writing Maker. And then, yeah, the last thing is probably the simplest part. Once you have your gene models and you're reasonably happy with those, you want to do functional annotation. I do have this slide here. So some of the things that people do to make the gene models a little bit cleaner is to remove things that look like transposons. So you can do an interproscan run, and that'll annotate proteins and let you know if they're potentially transposons. You might only want to take complete gene models. So Maker will actually allow you to have gene models that don't have a start and a stop. So you might be a little bit more specific there. You usually want to blast against known gene models, do some filtering with that information. FPKM, which I mentioned before, you can get that out of the string tie output. The AED score, the annotation edit distance. So this is a score that Maker will output that gives you an idea of how well the data supports the gene model. So if you have an AED score of 1, that means that the data doesn't support the gene model very well. So the lower the AED score, the better in theory the gene model. You can also compare to related species. As I said, Busco's really good. You can do gene families with OrthoFinder, and if you find your annotation, you have like a lot of unique genes relative to the other species you're comparing to. That might mean that you have to remove some repetitive elements. That's usually what that means. Oh, versioning, yes. So if you change anything in your annotation, everything should have versions. So like your first one might be, I don't know, 0.1, and then you make some changes and then 0.2. So just really important to have versions on your annotations, especially if you're doing a bigger project that involves a lot of people. And then, as I said, you can load things into Apollo and edit things to improve the gene models. Okay, I think that's it. Let me just look at that last script. Yeah, so this just shows you how to run InterProScan, which we downloaded at the beginning. So if you wanted to do that, you could. I think it should run in about 15 minutes or so once it's installed. And then this shows you how to do some of the runs against the different databases and anything that, and then you can just pull in those functions based on those matches to get some ideas of what the functions might be for your genes. Okay. Are there any questions? I know that was a lot. I'm losing my voice as I expected. I'm going to stop sharing my screen. Any questions?
What's the difference between the training thing and when you had a list of it going so many exons, so many protein coding, so many non-coding? Like, what's the difference? Because surely for it to give up that list, it had to be referencing to some kind of like guide file that informs it how to recognize what a protein coding gene might look like. So what's the difference between that and training it what a gene is or whatever that training thing
is? So I think I understand, but if I'm going off on a tangent, let me know. But so a lot of this is just creating the training set, I guess. So trying to get the gene models that are the closest to like biologically real as possible. So to get the training set, you want to have, you know, if possible, the start there. Any sort of splice sites should be as accurate as possible. And then hopefully you have the stop code on at the end. So all of that business at the beginning is just trying to get to that point where we have something that's pretty close to right. And then once we have that set that you know is pretty close to right, we just randomly break it apart into two chunks. So we take the first chunk, and that's what we use to train. So that's what's making the model for that particular genome based on the data that's saying, hey, this is what a gene looks like in this genome. And then once you have it trained, then if that training was good, it should be able to look at your test set and predict where the starts and stops and splice sites are in that test set. And since that test set you already created and you know where the stops and exons and everything should be, it compares to what you already know to what it predicted to get an estimate of how accurate it was.
So it compares it to an already described and defined genome or gene collection file thing.
Yep. So that's what all of the business with OrthoFinder in the beginning is. And basically, like before you get into the training, all of those steps are just trying to create that file that's as close to perfect as you can get it. So you're creating those. That's not something that like comes from somewhere else. You're creating that based on your own data. Some genomes you might already have some really well curated gene models, in which case you wouldn't need to do any of this. You already know some good examples of how, you know, a particular set of genes should look. So you could just go ahead and use that. But this whole workshop, we're kind of assuming that you just have some weird, you know, organism that, you know, nobody's worked with before, and you're just starting from scratch. So in that case, that's why you would need to generate your own RNA-seq and then do all of these steps to try to take that RNA-seq and come up with the best gene models possible. And then, you know, use that to go on to predict the rest of the genes, because you're throwing a lot of away in making these test sets. So that's why like your test set and your training set doesn't have all of the genes, it's just a subset. So once you have that Augustus train and SNAP train, then you go on and you know, attack the whole genome basically, and hope that that training set you made was good enough to, you know, more or less accurately predict genes in the entire genome.
But how do you choose what other species your test or training set or test set is from? Isn't it from a different species to the one that you're looking at?
No, it should. So you can do it that way, but in this case, I believe we use the same species.
But you don't. But wait, what? But isn't it supposed to be one that nobody knows what the heck anything's doing? So how can it say this is the proper gene? This is the proper gene format.
Yeah. So you wouldn't really know from a related species either, though. But that's why we do all of these steps in the beginning where we map. So we have the RNA-seq and we map that to the genome, and that gives us some idea of what the gene models might look like. So we do have some data, but the data is very messy. And we take all of these steps with all of these different tools to try to take that messy data and you know, hone it down into something that pretty accurately defines what those gene models should look like. But in doing that, we're throwing a lot of stuff away. So that's why the test set and training set, you know, all together might be, you know, only a few hundred genes, because you're only pulling out the ones that look really like high confidence, basically, in all of these steps that we did.
How does it know what a gene is? What, doesn't it have some sort of guidance of what a gene is?
From the RNA-seq. From the RNA-seq, and then whenever you run Mikado, you're also comparing against known proteins. So it's also taking that RNA-seq and comparing to a highly curated set of proteins that are from, that uniprot set is just from plants. So, I mean, that can be some pretty distant stuff. And I think a lot of it's probably from Arabidopsis. So that uniprot set doesn't necessarily work great for everything. But between looking at the RNA-seq, because the RNA-seq is mostly going to be coming from genes. So if you can figure out how that RNA-seq is mapping onto the genome and figure out, you know, from that where the splice sites are and where the starts and stops are located, and then you can start to figure out a training set based on that information.
With some information from previously described genes as well.
Mostly the RNA-seq, but then also we are using that uniprot set as well to in the Mikado pipeline.
But what about all the non-coding RNA that's floating around?
So in theory, I think a lot of that's probably not going to matter. Well, I mean, I don't know. I don't have exact numbers on this, but in theory, if it's coming from something repetitive, it should be masked out. So that shouldn't be interfering with any sort of gene model predictions. But if it's not repetitive and, you know, it's mapping to something, then depending on what your training set, I guess, told Augustus a gene model should look like, it may or may not call it a gene. Make sense?
I still just don't quite get how it knows what is right and wrong. But I'm probably going to take up too much time, so I'll probably find out some other place.
Feel free to reach out if you want to talk about this more. But I think the way, I mean, one of the ways that it's calling a gene, I guess, is just because you have the training and the test set. So that's what you're telling it is a gene. Like that training set, that you're telling it, this training set, the stuff in here, these are genes. So go and try to find anything else that looks like this.
That came from the thing that you don't know about?
That came from the RNA-seq, mostly.
Okay.
Yeah. And probably all this is a lot easier just using Breaker, and maybe that's in the future what we should consider doing, because Breaker, a lot of these steps you probably really wouldn't bother with. So Breaker is just taking the RNA-seq mapped to the genome, and then it's using that to, it still does training internally, but I don't think you really see much of it. And so it takes your RNA-seq and your mass genome, and then it gives you a GFF similar to what we just saw here.
Cool. Thank you.
And just to add a couple of things, because I love non-coding RNAs.
I actually thought of you whenever I started talking about it. I was like, oh, crap.
Yeah, I popped up my video so I could. So the idea with the RNA-seq is that you're trying to sample as diverse a tissue source as possible. But even so, you're still, I mean, and that's to capture as many genes expressed in the genome as possible. But typically when you do that, you're still only capturing the more highly abundant genes, which are typically going to be, in most cases, the protein coding genes. So those are the ones that are going to be captured in that training data sets. And those are going to be the ones that, you're going to call typically you're going to call other protein coding genes because of that. Link RNAs come later, long non-coding RNAs, small non-coding RNAs to a certain extent wouldn't be captured anyway in that RNA-seq, so you don't have to worry about them. But the long non-coding RNAs, those will come later after you've done a fairly decent annotation, and you can kind of get a sense of, there are other algorithms to capture those. So this is mainly going to capture the mRNAs, the protein coding genes.
Yeah.
What happens with the ribosome RNA?
Oh, ribosomes are so highly conserved. They're very simple to call. Those things, we could call ribosomal RNAs from an ant genome. Could take those ribosomal RNAs and call them in plants. They're that conserved. So there's like databases like RFAM and stuff that the annotations will scan against.
Thank you.
And yeah, if anyone wants to talk more about annotation, I know that was a lot in a very short period of time. Feel free to just shoot me an email and we can have a Zoom or whatever you'd like.